Hi, my name is Jeremy Linsley, and I'm a scientific program leader in Steve Finkbinder's lab at Gladstone Institute in San Francisco. Today, I'm going to speak to you about superhuman cell death detection with biomarker-optimized neural networks. Computer vision technologies have recently brought about major advances in our ability to harness computational power to identify patterns in images, such as through the use of deep learning-based facial recognition and detection, in which a particular person's face can be recognized from amongst thousands of others. As scientists, I think it's fairly straightforward to think of the many ways this technology could be used to boost our ability to make discoveries in biomedical science. Yet in reality, practical implementation of these technologies into biomedical research has been limited due to two major technical limitations. One is that deep learning often requires very large labeled data sets to train deep learning models. This problem can be partly overcome through the use of automated imaging technology. In the ThinkBinder lab, we have developed several different platforms we call robotic microscopes, shown in the video here on the left, that conduct automated longitudinal live imaging of cells and imaging plates that are returned to incubators after imaging to keep them alive for up to months at a time. This technology allows us to easily generate hundreds of thousands of images of cells per day. However, each cell must still be typically curated or annotated. It's a costly and slow process that also introduces human bias. The second problem is that complex calculations and convolutions carried out by deep learning models are often considered a black box that is uninterpretable. This means that although they can often generate fantastic results, such as correctly predicting the ICC staining in a bright field image, they give no information on why or what caused the pixel to be predicted as such, which often leads to scientific dead ends. In the ThinkBinder lab, we mainly employ robotic microscopes to carry out live imaging experiments of neurons undergoing neurodegeneration over long periods of time. With the growing and almost endless library of live biosensors available, such as mitochondrial structure and function, organelle trafficking, calcium activity, or proteostasis sensors, we can then monitor the physiology of neurodegenerative disease models to identify antecedent pathways and therapeutic targets that can be modified to stop neurodegeneration. However, one problem we typically encounter in these experiments is the inability to detect the precise timing of death of neurons that we are imaging. It is often very difficult to distinguish in fluorescently labeled neurons between signs that a neuron is simply unhealthy and signs that a neuron has reached a terminal endpoint from which it will never recover. This is particularly problematic because we often find fluorescent debris can persist in culture for up to 48 hours after a cell's death, even though we are only interested in the biology of the neuron before it died. In response to this challenge, I developed a novel biosensor to detect neuronal death. Based off of a genetically encoded calcium indicator, this novel biosensor, which we renamed Genetically Encoded Death Indicator, or JEDI for short, is specifically engineered to only detect extremely high levels of calcium within neurons that are indicative of irreversible commitment to death. We find this indicator provides unprecedented resolution of time of death of neurons in live imaging experiments. By fusing a red fluorescent JEDI indicator to a P2A cleavable peptide and a complementary EGFP cassette, we were able to generate a quantitative ratiometric fluorescent signal that robustly differentiates between populations of live on the bottom of this graph and dead neurons on the top, and it's amenable to high throughput imaging. Each neuron captured during imaging can then be cropped to a 300 by 300 pixel image centered around the soma and automatically sorted as dead, which looks yellowish on the left of this image, and green and live, which looks greenish on the right here. Repeating this process in an automated pipeline allows us to easily and automatically generate essentially endlessly large data sets of live and dead neurons. But although JEDI gave us a robust live dead signal, imaging with JEDI took up two fluorescent channels, limiting our ability to concurrently image other biosensors at the same time. So we next wondered if we could train a convolutional neural network model to detect death from the EGFP morphology of the neuron alone, allowing us to bypass the use of JEDI to determine death in other live imaging data sets. We paired the JEDI-derived classification label with EGFP alone images and trained a convolutional neural network, or CNN, to classify images as live or dead based on the morphology of the image alone. We found that a successfully trained JEDI CNN model showed over 96% accuracy in detection of death in live longitudinal imaging experiments. And in comparison to human curators, which were previously thought of as the gold standard for detection of death in live imaging experiments, the JEDI CNN showed superhuman accuracy and over 100 times the speed of a human. Not only did the JEDI CNN translate to new imaging data, it also showed superhuman or human level accuracy in detecting death in neurons from other species, such as mouse neurons, or different types of neurons, such as human iPS-derived motor neurons, and incredibly, even in cells that are not neurons, such as HEC293 cells. In order to gain an understanding of the basis of the classification decisions by the Jedi CNN, we next use guided gradient-weighted class activation mapping, or GradCam, to generate a quantitative mapping of the pixels underlying the Jedi CNN decision. We found that the GradCam signal clearly localizes to the contours of live and dead neurons. It can be co-localized with different biosensors of subcellular co-localization, such as CAX-M-APPLE, shown here, which labels the membrane of the neuron. When we compare the subcellular localizations of the GradCam signal of live to dead neurons, we find that the GradCam signal in the soma of neurites is reduced when a neuron dies, which makes intuitive sense because the membrane and neurites of neurons are well known to decay as the neuron dies. But surprisingly, we found that the GradCam signal within the nucleus of a neuron also appears to be increased in dead neurons compared to live neurons. This was particularly surprising because our eyes have difficulty detecting any type of pattern in the nuclear corresponding area of morphology of dead images. To better characterize how different subcellular signals drive classification, we next generated a series of pixel ablations corresponding to different subcellular regions of each neuron of live or dead neurons to see which pixels were necessary for the Jedi CNN's decision. We found that ablation of the GFP morphology pixels corresponding to the neurites and soma membrane changed the classification of live neurons to dead, which was to be expected. Conversely, and unexpectedly though, we also found that the ablation of the GFP morphology pixels corresponding to the nuclear signal changed classification of dead neurons to live, but did not change live neurons to dead. This suggested that the Jedi CNN used a previously unrecognized and non-obvious signal within the nucleus of the cell to detect death. In line with this observation, when we subdivide the data into images in which consensus of human curators were inaccurate at classification compared to the rest of the data set, we find that the CNN was especially dependent on the nuclear signal to generate its correct dead classifications where humans were unable to make the correct call, suggesting the nuclear signal underlies the superhuman accuracy in detection of death. To summarize what I just told you, we used a biomarker-optimized CNN strategy to generate superhuman and translatable live dead classification accuracy without the use of curation. I then showed you that the resulting Jedi CNN can be interpreted on a subcellular level using GradCam, and that the interpretation of the Jedi CNN signal can lead to the novel biological insight that the nuclear region of the EGFP morphology contains information that can indicate neuronal death. With that, I'd like to thank all of these people, and I'm happy to take any questions. Thanks.